Hello, Warrior Cats fans and other people writing about or drawing fictional cats. My name is Murphy, and I have a degree in, evolutionary biology, but I have a strong background in classical genetics, and I specialize in domestication. I see a lot of people in the Warriors fandom wanting to make their cats, or the book's cats, more realistic by incorporating real-life genetics into their cats, and that's great! But I also see a lot of people doing it wrong. Now, don't get me wrong, creative freedom is great, and no one should be bound by the reality of genetics against their will. But if you want to get as scientific as possible, you've come to the right place. This is going to be a series of videos deep diving into the genetics of cat appearance. It may be tempting to skip ahead and watch a specific video for a specific gene, but don't. These videos will be simplified and non-comprehensive, with non-scientist artists in mind, but despite that, I believe it's important to not just know the surface level of what you're doing, but to understand why and how it works. Thus, future videos in this series will expect you to have a solid foundation in both genetic theory and jargon. That's what this video is for. This is your primer to understand the rest of the series. Welcome to Cat Genetics for Writers and Artists, Part 1, Concepts and Terminology. By definition, genetics refers to the study of particulate inheritance. What does that mean? Well, Gregor Mendel is credited with discovering genes and thus is considered the quote-unquote father of genetics for his experiments involving breeding pea plants. Before him, it was generally believed that heritable traits were passed down by blending traits together, like blue plus yellow equals green. People look like a mix of their parents. What Mendel discovered was that genes actually don't blend, not at all. When he bred green and yellow peas together, he got green peas. Then when he bred those green offspring together, like magic, he got yellow peas again. This suggested that something inside the peas was remembering the parent's traits instead of smushing them together or discarding one. This is what we mean when we say particulate inheritance. There's a physical particle inside of the body that knows your heritage and can be inherited by your offspring. It wasn't until much, much later that we discovered what those particles are. This video is going to be in-depth, detailed, and complicated, and it will not even scratch the surface. You do not need to memorize or even fully understand everything that I say. You don't need to be an expert on genetics to write and draw more realistic cats. But genetics is an important field that studies very real phenomena that affect all of us. Just like history, even if you only study it as a fun hobby, you have a responsibility to understand and acknowledge that the reality is much more complicated. Throughout history, genetics and inheritance have been used as tools to discriminate against and put people down. Misunderstandings of genetics have led to especially heinous crimes in the last century, from justifying racism, sexism, and transphobia, to the forced sterilization of Emma Buck, to pro-genocide propaganda during World War II that continues to this day. It would be irresponsible of me to make any kind of educational content on genetics without quickly and decisively dispatching the most common mistake that leads to bigoted misunderstandings of genetics. Genes don't actually code for traits in the way that people think. What genes code for are proteins. Here is a chart of what genes actually code for. Note that precisely none of these say intelligence or beauty or even attention span or nose shape. They're things like leucine, alanine, lysine, etc. These are proteins that, when you get a lot of them, work together to build the more complex systems that make up your entire body. Different versions of a gene often, but don't always, lead to different traits, not because one trait is being swapped out for another, but because one or more proteins are being swapped out, or moved around, or prevented, or a number of other things that could change how the proteins are created. Traits are more complicated than we think of them as being. You might think of your hair as just curly or straight, but the reality is that millions of tiny details like the shapes and sizes and density of your follicles, oil production, testosterone levels, things like that all affect your hair texture. Changing any one of those tiny details changes your hair texture. Genes reflect this complexity. They affect the building blocks of traits, often in minor ways that logically result in differences that we see, not straightforward one-to-one blueprints of a whole trait. Sometimes those building blocks build multiple things, so a gene can have multiple seemingly unrelated effects. For example, a gene that changes hair follicles, which both grow hairs and are found in the skin, may cause both a hair trait and a skin trait. Genes that do multiple things like this are called pleiotropic genes. Inside your cells, and cat cells, is an organelle called the nucleus. Inside that nucleus is your genome, all of your genes wrapped tight around spools called histones. This is the chromosome. Chromosomes have two strands, two backbones that are held together by this ladder rung structure in the middle. The ladder rungs are actually two different molecules tightly bonded together called base pairs. Those base pairs are genetic information. That's the genetic code. Chromosomes exist in pairs. Chromosomes that belong in the same pair are called homologs of each other. Homo meaning same, like in homosexual, attraction to the same sex, or Homo sapiens, same species as us. When the body produces sex cells, eggs and sperm, both called gametes, it separates all the homologs. So each gamete only has one of each chromosome instead of full pairs. Egg and sperm combine to create a new complete genome. The separation process, meiosis, is fascinating and complicated, and outside of the scope of this video. The only important thing for you to know is that before it happens, homologs will swap trunks of genes with each other. So you aren't getting the same exact chromosomes your grandma gave your mom, for example. Some of your grandpa's DNA is in there too. Important side note, I'll sometimes use the terms mom and dad in these videos because it's easier than saying egg donor and sperm donor every single time, but gender is a psychological field, not a genetic one. Trans rights. The important takeaway here is that you get one set of genes from your mom and one set of genes from your dad. They exist simultaneously within the body as homologs. The exception to the rule of chromosomes being in pairs are sex chromosomes, X and Y. In most AFAB individuals, this will be a normal homolog, XX. But in most AMAB individuals, there's only one lone X and a Y. Some people have more than two or only one copy of certain chromosomes, which results in various disorders, many of which are survivable. You don't necessarily need to know the names for different kinds of mutations, but I want to cover the term mutation itself, because it's another often misunderstood term in science. A mutation is when some part of the genetic code changes, usually because cell division made a mistake. At the beginning of life, we were single-celled organisms with only RNA and no DNA. Then we got DNA, and that DNA has been mutating ever since. Every change in the history of evolution leading till now is a mutation. Thus, all current genes are mutations, all of them. Even if the change itself happened generations ago, the resulting gene is still called a mutation. Some mutations are more recent, speaking in broad evolutionary time, like blue eyes. But even having eyes at all is the result of countless mutations over countless years of evolution. The term wild type is used frequently by geneticists to refer to quote-unquote original or the most common mutations that a species has to differentiate between old mutations and new ones. In cats, we might say that this mackerel stripe pattern is a wild type because it was the original cat stripe pattern they inherited from African wildcats at the dawn of domestication. The takeaway here is that when I say mutation, I'm not talking about cats who are suddenly mutating these genes or that they're horror movie mutants. I'm just saying that it's a gene that won't be found in African wildcats. So here's something that's gonna mess you up. The word gene is kind of outdated. It was coined long before modern understandings of genetics. The definition of gene is a unit of inheritance, the particle and particulate, if you will, which it turns out there's a lot of. The two words genetics will use in place of gene are locus and allele. Mind, we do still use the word gene, but really only when it's clear which of the two we're referring to, or when specifying doesn't matter, like me in this video so far. The locus is the location of a gene. Plural is loci. This is the physical part of the chromosome where a gene is located. When people say homologs are identical, they mean that they have the same loci as each other, information all organized in the same places. What that information is called the allele. This is the actual code that leads to specific proteins. I like to think of it like this. Here's a street. All the plots along the street always have the same address. 123 Jean Street is next to 124 Jean Street is next to 125 Jean Street, and so on. But what house gets built on that plot can vary. Modern, old Victorian, country style, etc. This is why the word gene can get confusing on its own. When you say the gene for stripes, do you mean the locus that would determine whether stripes are present or not? Or do you mean the code for stripes itself that you would find on that locus? Because every cat in the whole world has the locus for stripes, the part of the chromosome that determines if a cat is striped or not, but only some cats have the allele for stripes. Remember this chart? What do these letters mean? Those are base pairs. Remember base pairs, the ladder rungs holding chromosomes together? Well, technically these letters are only half a base pair. They're called pairs for a reason. Now, obviously, there aren't any actual letters inside your cells like you're a can of alphabet soup. These letters represent the molecules adenine, thymine, guanine, and cytosine. We just shorten them to A, T, G, and C. To make the rungs, A and T pair up, as well as C and G. But RNA can only read one half, so when we talk about the genetic code, we only need half the latter. 3 nucleobases together, like we see on this chart, make up a codon, like letters forming a word. RNA reads them together. It takes a lot, and I mean a lot, of codons to code for any given trait. So when we say that a trait only has one gene associated with it, like stripes, that just means we only need to check one locus to see if the right allele is there. But that allele is probably huge, and within it there's promoters, enhancers, inhibitors, introns, and all sorts of things that get spliced and glued by RNA before it becomes a protein. One locus, with heavy air quotes, can code for several different things if read and cut differently. All you need to know is this. This street. A locus is a location. An allele is the information on that location. Different codons, different allele, same locus. Loci and alleles are often represented as letters. Don't get confused, this is different from A, T, C, and G. They don't represent specific molecules. They're shorthand for traits, chosen somewhat arbitrarily, often by breeders before the genes behind the trait are even understood. We aren't going too deep in any specific genes today because everything's going to get its own video in the future, but to give an example, the B locus is the part of the cat's genome that tells us whether a cat is black, chocolate, or cinnamon. The alleles for this are capital B, little b, and B1, respectively. When we then write the genotype for a cat, meaning the alleles that they have, we'll write them like this, 2 letters. One B represents information from one homolog and the other from the other homolog. I see people write them without the slash, and that's fine too. Whether it's less professional or not depends on who you talk to. One of my professors was pretty strict about the slash, so I put it there, but you're not wrong for not including it. Now, an individual organism only has 2 alleles in their genotype, one for each homolog. When we zoom out and look at the wider population, we can take all of the alleles at the same locus of everyone's genotypes and put them into a big imaginary pool called a gene pool. And here we can see there's a lot more than just 2 alleles. So one cat may only have 2 black alleles, but a different cat only has 1 chocolate allele and 1 cinnamon allele. Thus we can say that this locus has 3 associated alleles, even if each cat only has 2. When someone has 2 copies of the same allele, we call them homozygous. Remember, homo means same, like homosexuality. And when someone has 2 different alleles, that's called heterozygous. Hetero meaning different, like heterosexuality. And finally, when there's an XY individual, any alleles on the X chromosome have no pair. It's just one lonely letter all by itself. That's called hemizygosity, with hemi meaning half. So genotype refers to what alleles a cat has. Meanwhile, phenotype refers to what the cat's traits actually are as they appear on the body. Remember Mendel's pea plants? The ones that have the green phenotype, but their genotype contains both green and yellow? So why are they green instead of yellow, or both? The answer is dominance. This is another concept that gets bastardized by misunderstandings and can lead to a lot of suffering if misused. The word dominance is misleading, and it has a lot of implications in Western society. Alleles can either be dominant or recessive. Dominant alleles don't cause a person to be more socially dominant, and they don't exercise any kind of anthropomorphic power dynamic over recessive alleles. Dominant alleles get read instead of recessive alleles. That's all it means. A better term might be high priority. Dominant genes also aren't more common. The human gene for 6 fingers is dominant. Genes are more common when they're beneficial to reproduction, or when we breed for them deliberately, not when they're dominant. Oh, and when we write alleles that someone has on their locus, we write the dominant allele first, like this. Typically, dominant alleles are capital letters, and recessive alleles are lowercase letters, but sometimes we use numbers, or superscripts, or multiple letters. Dominance is also relative. Alleles aren't inherently dominant or recessive. Rather, alleles associated with a locus have a dominance order. Let's take a look at the black locus again. Uppercase B, little b, and B1. Little b, which codes for chocolate, is recessive to capital B, but dominant to B1. So if the cat is capital B, little b, chocolate is recessive and doesn't get read. The cat is not chocolate. But if the cat is little b, B1, chocolate is dominant and it gets read, and the cat is chocolate. The allele that gets read is expressed in the phenotype. The allele that doesn't get read is carried. There's also 3 different types of dominance. The type I just described is complete dominance, wherein the dominant allele is read and the recessive allele is not. The other 2 are codominance, where both alleles get read but in different places, expressing a cool mosaic phenotype, and incomplete dominance, also sometimes called semi-dominance, where both alleles get read in a way that overlaps, expressing an in-between phenotype. When we write dominance orders, we write them like this, with the greater than sign referring to the more dominant allele as greater. If the genes are codominant or incomplete dominant, we use an equal sign, like this. As mentioned before, XY individuals are hemizygous for every trait on the X chromosome, meaning they only have one allele. Since there's nothing to compare it to, the lone letter isn't dominant or recessive. It always gets read. This leads to something interesting. Some traits show up more often in XY folks since they have nothing to compete with, and some traits don't show up at all because they require co- or incomplete dominance to show up. This is why more males are colorblind than females. Most forms of colorblindness are recessive to full color vision, but they're also located on X, so it can freely express in XY folks. This is also why male torties and calicos are rare, but that's a future video. Now that you understand how to read and write an individual's genome, it is finally time to learn how to calculate offspring. If you took high school bio, you've probably seen this before, and you might remember it's called a Punnett square. This is a very simple mathematical equation that will help us see every possible genotype that a pair can produce in their offspring. Let's go over how to solve it. Take any two individuals and pick one locus. Here I have a black cat who carries chocolate and a chocolate cat. Put their genotypes on the top and the side of the Punnett square like this. It doesn't matter which one is mom and which one is dad. Personally, I put dad on the top and the mom on the side just so that I don't forget which one is which, but it does not matter. The results will be the same. This represents the alleles getting split up into different gametes. If it helps to imagine, you can extend this line inside the square to further separate the alleles. Bring the letters from the top straight down, filling the boxes below, like this. Then bring the letters on the side out, filling the boxes on the right, like this. Now all four squares have a complete genotype. A common misunderstanding is that this represents 4 kids, or a litter of 4. Someone might see this and say, okay, half the litter's black and half the litter's chocolate, but that's not correct. This actually represents 4 different possibilities for the same kitten. Essentially, the Punnett square simulates 4 alternate universes where different gametes combine with each other in 4 different possible ways. In this case, in 2 of the 4 universes, the kitten is born black. Since 2 is 50% of 4, we say this has a 50% chance of happening. Same for chocolate. Of course, if they have the same parents, you only need to do this one Punnett square for the whole litter. Just don't feel like that litter has to be split down the percentage lines. Due to how chance works, you will most likely see whole litters lining up roughly with Punnett square results, but just like a coin flip, sometimes you just get a bunch of heads in a row, and a 50-50 litter ends up fully black. You're a writer or an artist working on fiction, so have some fun with it. Maybe a single black protagonist amidst his all-chocolate litter mates. Get creative. Let's do another more complicated example with Mendel's pea plants. Here's 2 green peas that both carry yellow. Separate their gametes by putting the alleles on and beside the square. Great! Now bring them down, and bring them out. Now this is interesting. Remember, green is dominant, so in 3 out of 4 results, the baby pea is green. And in only 1 of those 4 results, the baby pea is yellow. This means the pea has a 75% chance of being green and a 25% chance of being yellow. That's calculating for phenotype. The 75% and 25% there refer to the baby pea's actual expressed appearance. Calculating for genotype instead, we can see that 2 out of 4 results have a heterozygous baby pea. It's carrying yellow, so the baby pea has a 50% chance of being heterozygous. One result has 2 capital G's, so that's a 25% chance of homozygous dominant, and the last result has 2 little g's, that's a 25% chance of homozygous recessive. If the fancy percent numbers are scaring you, don't worry, just Google what percent of 4 is 1 and you'll be able to skip the math. Punnett squares look the same for sex-linked traits, except for the hemizygous XY parent, a different symbol is used to represent the lack of an allele. I personally use a Y, but one of my professors used a dash, another one used NA. As long as you know there's no allele, you can put anything. Put a smiley face. Draw a P. Most Punnett squares are done for a single locus, which means you'll have to do a lot of them to figure out the full appearance of even a single kitten. Personally, I think Punnett squares are fun, so I don't have a problem with that. But sometimes you want to go a little faster. Maybe do 2 or even 3 loci in one go. I'm happy to inform you that you can! Punnett squares can be done with multiple loci at once, but they get far more difficult. If you're happy with very simple math and you don't mind drawing a lot of Punnett squares, skip the next section. If I did my job right, there should be chapter select for this video, but if you're like me, and you really like math, and you want the advanced strats, I'm happy to introduce you to Multi-Loci Punnett squares. Okay, so the hardest part is separating the gametes. Here the loci I'm using are the B locus, which we've gone over, and the agouti locus, which is the tabby gene. Capital A for tabby, little a for solid. Got it? Got it. Dad is a black tabby, aka a brown tabby. I'm just gonna call him a black tabby for this. Mom is also a black tabby. Dad is homozygous black, but he carries solid. Mom is heterozygous on both loci. Let's start with mom, because she's going to be the easiest. Meiosis is going to duplicate her chromosomes and then separate them out into 4 gametes. Separate the B's and the A's. Now make every possible combination of B's with A's. Not B's with B's, not A's with A's, just B's with A's. We have capital B, capital A, lowercase b, lowercase b, capital A, and capital B, lowercase a. Those are all 4 gametes that mom might give to baby. Now it's time to turn this Punnett square into a Punnett rectangle, because dad has some homozygosity. Let's make his gametes. Capital B, capital A, capital B, lowercase a, and that's it. There's no lowercase b, so those are the only possible combos. Now, you can, like with the single locus squares, write big A, and big B, little a twice each, but it takes longer, and it takes more space, and for me at least, it makes the math harder. Now, our Punnett square rectangle looks like this. Now we do the same thing we did in the single locus square. Bring one parent's alleles down, and the other parent's alleles over. Again, it helps if you mentally separate the gametes by drawing the lines further up and out. It helps to write one parent's letters on top of each other, like this, because it gives you the space to put the others in correctly when you get to them. I'm going to fill out the rest. Feel free to pause and try to do it yourself. Okay, now for the other parent. Again, feel free to pause and do it yourself. Now we have all possible alternate universe genotypes for the kitten completed. It's math time. Whether we're calculating for genotype or phenotype, the first thing we're going to do is count how many squares there are in total. We have 8. Next, we're going to find identical universes. If you're calculating for genotype, group together all the squares where the letters are exactly the same. If you're calculating for phenotype, group together all the squares where the kitten looks the same, regardless of what recessives they're carrying. I'm going to calculate for genotype first. Find all the genotypes you're interested in, and then count how many squares have that exact genotype. Let's go with full heterozygote. Capital B, lowercase b, capital A, lowercase a. I see 2 of those. If you're having a hard time parsing all the letters to find identical squares, here's a trick. Draw a different arbitrary symbol in the corner of the square for each unique genotype. Same symbol equals same genotype. I do this for the whole Punnett square before I start on any calculations, but you don't have to. Here's what mine looks like after I drew the symbols in. The 2 full heterozygotes are marked by that little circle. If you had a hard time seeing them amidst all the letters before, it should be more clear now. That's 2 out of 8 squares, so use a calculator or just Google what percent of 8 is 2, and you'll see that this pair has a 25% chance of producing a full heterozygote kitten. Same process for when a genotype is totally unique. The full dominant homozygote up there with the plus symbol is all alone. So what percent of 8 is 1? This pair has a 12.5% chance of producing a fully homozygous dominant offspring. At these 2 loci, anyway. Now let's calculate for phenotype. This one is where those arbitrary symbols become even more useful. I use the same symbol on squares that express the same phenotype, regardless of what they're carrying. You can also write all the possibilities off to the side and then tally. I'm going to do both. Pause if you want. We have 6 black tabby possibilities and only 2 solid black possibilities. I also wrote chocolate tabby and solid chocolate off to the side, since one of the parents was chocolate, so someone less experienced might have expected a chocolate kitten. But as you can see, Dad's homozygous black made that impossible. There's no chance for a lowercase b homozygosity. The most it gets is carried. Congratulations! Now you can do Punnett squares with as many loci as you can fit on your piece of paper. Now back to the basics. Traits that are controlled by a single locus are called monogenetic traits. Traits that are controlled by multiple loci are called polygenetic traits. Polygenetic traits can be controlled by 2 loci, 3 loci, a few dozen, even thousands of loci. The vast majority of traits are polygenetic in nature. Monogenetic traits are few and far between. We already went over monogenetic traits in the Punnett square section. 1 simple locus, 1 simple square. There's a lot of different kinds of polygenetic traits, though. When someone says a trait is polygenetic, they're usually referring to a gradient trait. Gradient traits have so many loci working together to make them that there is a ton of slightly different phenotypes. If you line them up, they'd look like a gradient. This usually works, in simple terms, by each locus acting as a plus, minus, or neutral to the total result. Human skin color is a gradient trait. Humans aren't just dark or light, there is a wide spectrum. The genes responsible for skin color each either add or subtract a small amount of melanin, a black pigment. More melanin means darker skin, less melanin means lighter skin. Together, a large amount of genes adding or subtracting small amounts of melanin result in the final skin color. Often, we don't know all of the genes behind gradient traits. Sometimes we don't even know one. Genes are really hard to find. It's fine if you're calculating gradient traits to just assume the offspring is going to be somewhere between the parents. It's also possible for a kid to end up more towards either end of the gradient than either parent. This is really important in evolution, but that's not what this video is about, so just know that it's possible. The other really common kind of polygenetic trait is called a complex. This is when there's a primary locus that determines if a trait is expressed or not, and then a bunch of other loci, called modifier genes, work together to determine how it's expressed. I forget what the actual word is for the activator locus. I call them light switch genes to myself because it's like determining whether the lights are on in a room. If the locus has off alleles, the room is dark and you don't know what's in it. But if the locus has on alleles, you can see everything in the room that all the modifier loci put there. My favorite example is the leopard complex in horses. Leopard gives horses a cool white marking, but that white marking can look super different depending on what the modifiers are doing. These horses are all leopards. They all have the same on allele at the same light switch locus, but their modifiers are different. Tabby in cats is also a complex. There's a light switch gene to determine whether the cat has stripes or not, and then a lot more genes determine what the stripes look like, but more on that in a later video. Earlier, I said we already went over monogenetic traits in the Punnett square section, but technically just about all monogenetic traits are technically complexes. There's a lot of different shades of chocolate, for example, but if we don't know the modifiers, and there's a lot of them, and they don't really make that much of a difference, we just pretend it's a monogenetic trait, we can use a single Punnett square and get expected results. Some traits seem like complexes, but are actually determined not by genes, but either by chance or other external factors during development. Even if a trait is not determined by genetics, we still use the word phenotype to refer to how it appears on the body. Quick example, if two fetuses combine in the womb and make a chimera, we say that their phenotype is chimera, even though genetics didn't do that. The last term this video is covering is epistasis. Put simply, epistasis refers to when an allele can't express because it's blocked by a totally unrelated allele on a totally different locus. You'll recall that genes don't actually code for black or stripes, they code for a bunch of proteins, and it takes a lot of them to make a trait. Just like waiting on your group project partner to email you the damn file already so you can do your half of the project, one gene can't do its job unless the other one does it first. If your partner never mails you that file, the project never gets done. You might have all the genes of a beautiful, many-colored cat, and then one bastard doesn't do its job and now the cat is entirely white. Whoops! Congratulations! If you made it all the way through this video, you now hopefully have a baseline understanding of important genetic concepts and a working genetic vocabulary. You should now be able to understand what I'm saying when I use jargon in later videos. But, if you need it, there is a link in the description of this video with a list of key terminology. Genetics is difficult, and you should not feel bad if you can't memorize the whole video in one go. I'm condensing nearly my whole life of independent genetic study plus a college degree into this thing. Please, Google terms you don't remember. Use the doc I linked. It's okay to need help. This isn't a test, and I just want to help artists who are interested in realism form a deeper understanding of a fascinating field, as well as dissuade dangerous misconceptions that they may come across. Later videos will be shorter, and I'll cover specific loci or related groups of loci, and will be much more cat-focused. I'll see you there.